would now like to introduce Pantelis Vlahos, Principal Strategic Consultant at Cytel.
Hi, everybody. Thank you, Alyssa. And thank you, everybody, for joining this webinar in these very challenging times. This is the first of a series of 5 webinars that we'll be presenting on behalf of Cytel, covering different topics that are of interest to statisticians. We are very eager to get your feedback on this and obviously to expand by offering more topics in the future. So, as Alyssa mentioned, and now I'm just going to switch to the presentation, I will cover today the topic of adaptive umbrella trial design using the MAMS module in EAST. In all of the modules and all of the webinars, this weekly series for the next 5 weeks that we're going to be covering, there is going to be the similar type of structure like the one that you're seeing today. So there's going to be some background on the methods and how we're going to be implementing that method in EAST, and we're going to be illustrating that implementation through a case study that we're going to be covering each time. So this is the structure that we have here as well, a little bit about the methodology, the case study, any questions that you have, the conclusions from today's webinar. So EAST, for those of you who are familiar with it, or even for those who are not, has a variety of modules that typically tries to meet all your design needs. You can see them here. It started in the late 80s with a version that would cover the design of group sequential studies, as well as which is covered currently in the sequential module, as well as for normal, as well as for binomial and time to event endpoints. It then expanded to include several adaptive features with the use of the adapt and the serve adapt module. But from that point and onward, we started adding, we scanned the literature, tried to keep abreast of the methods and try to cover the design needs of statisticians in the industry by including methods for early phase, like the escalate module, phase 1-2, like the MCP module, which is one of our latest addition, phase 2-3 designs, which is part of the MAMS module that I'm going to be covering today, and confirmatory designs, confirmatory group sequential and adaptive designs, as well as specialized designs that deal with multiplicity, either multiple arms compared to control or multiple endpoints. So I'm going to try through these 5 weeks to give you a flavor of some of these, although today the emphasis is going to be on MAMS, which is our module for multi-arm, multi-stage clinical trials. And it has as a prerequisite 2 other modules in EAST. One is the multi-arm module, which enables you to design fixed trials whenever you have multiple arms compared to control. And it also requires the sequential module, which allows you to design group sequential trials. Because as you will see, one of the issues that we're going to be showing today is that how you can extend actually group sequential design, a standard 2-arm group sequential design, in a case when you're going to be having more than 2 parallel arms. So the screen that pretty much describes the functionality of the MAMS module, all of the functionality of the MAMS module, is the one that you see here. And of course, these slides are going to be made available to you as well at the conclusion of the webinar. And it currently includes 2 different methods, although today we're going to be covering one of them. The first method that includes is the 2-stage treatment selection method, with the p-value combination, which was developed by Posserol in 2005. This is a standard design that has been used typically for inferentially seamless phase 2-3 designs. So with this approach, you have multiple arms compared to control, you have an interim look, and then at that interim look, you have the opportunity to graduate into the second stage of development the best or the two best treatment arms along with control, and then combine the results of the two stages with a p-value combination function. This is part of the functionality of VIST, although we're not going to be covering that. Today we're going to be seeing the group sequential extension to more than two arms, which will lead us to identifying promising therapies, compare multiple treatments at a time with control, and allow several primary research questions to be answered in a single study. So let's talk a little bit about the methodology and about also the type of trials that lend themselves naturally to this methodology. So these type of trials result from the use of master protocols, where by master protocol we mean an overarching protocol or a trial mechanism which comprises of several parallel sub-trials, which typically differ by several molecular features or maybe some other objective. There are different flavors of master protocol trials. You probably have heard already of the basket trials, where you have a master protocol where each one of the sub-trials enrolls multiple tumor types, and that is what we call it, the basket. There is the umbrella type of trials, where all patients and all sub-trials share a common tumor type. That would be the umbrella. And then we have a platform trial, which where the sub-trials may be added or removed in an operationally seamless way. As we mentioned on the title of today's presentation, our emphasis will be on the umbrella trials, and there are different implementations of those trials as well. In this very seminal paper by Woodcock and Lavange, where they presented master protocols, they looked at this type of umbrella trials, where you have a single disease, there is a screening for the presence of different targets, presence of different biomarkers, and then depending on what sort of biomarker is present, there's going to be a different targeted therapy assigned. So this particular example here does not have a control group. Our extension, what we are going to be presenting today, is a case where there is actually going to be also a common control in the study. So the motivation that we have is, of course, how to deal with the umbrella type of designs. We want to identify promising therapies and perform inference on the treatments that we select, and not just confined on two stages like the p-value combination approach, but also extending it into more than two phases. And we're actually going to be illustrating that today with a case study that we have. We have multiple treatments compared to control, and several primary research questions can be answered in this single trial efficiently, more efficiently compared to having separate trials. There are different modifications that lead to distinct MAMS designs. There are group sequential MAMS designs, drop the loser designs, multiple winners designs. We're going to try and see as many as possible of these. As I said, our emphasis today is going to be on the extension of the group sequential design. So this is a case where, again, we compare more than one treatments against the common placebo, and we extend Dunnett's test essentially to multiple stages. At any interim look, we have an opportunity to stop the study for efficacy or for futility. So as this is an extension of a group sequential design, that means that we construct efficacy and futility stopping rules based on known stopping spending functions. So we can stop the study early for efficacy if any of the treatments crosses the efficacy boundary, and we can stop the study for futility if all of the treatments cross the futility boundary. So if all of the treatments are significantly worse than placebo. If only some of the treatments cross the futility boundary, then these treatments can be dropped from the remainder of the study, and the trial may continue to test the remaining treatments. As we are using spending functions and we control how to how alpha and also the type 2 error is being spent if we have a futility stopping rule through the study. There's no need for close testing to maintain the strong control of the family-wise error rates. This is something, of course, that is needed in the p-value combination approach by Poserol. Mathematically, here's how we think about things. If you look at the very top of the screen, we have the standard K-look group sequential design where you have one experimental treatment compared to control. And you have this comparison made K times. These are the different interim looks that you have, as well as the final analysis. The way that we find the efficacy boundaries is by controlling the type 1 error, by imposing the constraint that under the null hypothesis, the probability that we will stop either on the first or on the second or on the kth look will be equal to alpha. This is extended in the case of d-comparisons to a common control. Now, the only difference is that instead of looking at these scalar score functions and trying to see whether or not they cross the efficacy boundary, we are looking at the maximum of their values of this d-dimensional vector, which has the d different treatment arms. And we want to see what is going to be the first time that it is going to cross the efficacy boundary, so that under the null hypothesis, the probability of this happening will be equal to alpha. If we were to graphically depict this in the two-arm case, the standard group sequential approach, where we have the score functions W, these are scalars, the trial stops either at the first look if W1 is above E1, or at the second look if W2 is above E2, or of course at the final look if W3 is above E3. And we want the chance of this happening under the null to be equal to alpha. The computations are simplified because we have the independent increment property for the score functions. On a multi-arm trial, now the score functions are actually vectors. And now we cannot interested at the individual components of the vectors, but we're interested at the maximum components. And the trial will stop if the maximum component of the first look is greater than your efficacy threshold, or if the maximum component of the second look is, or at the third look is going to be. And again, what we want is that under the null hypothesis, the chance of this happening will be equal to alpha. Computations now are a little bit more complex because the components of the vector itself are correlated, but we still have the independent increment property between the vector and, of course, the increment to get to that vector. So this is, in a nutshell, what's sort of like the theoretical background behind this. This is what is involved when we want to calculate the efficacy boundaries. For the futility boundaries, the approach is a little bit more intense because it requires first finding the efficacy boundaries, fixing a value of a futility boundary, and then having an iterative procedure to find the futility boundaries, which would require multiple integrations over different distributions, in this case, of course, multivariate normal distributions. And this is quite time-consuming. It has been shown by different publications in the literature that, for example, if you're dealing with a four-arm design with four stages, 4 looks, it might take up to 8 hours with R to compute the futility boundaries. Such computations, we have managed to bring them down in the order of minutes, and actually you will see live how we can deal with such designs and how quickly something like this can be performed. So we're going to be illustrating our designs with a case study, which is going to be taken from the area of COPD. So we have once daily bronchodilators for COPD, and what we do is we compare 3 different doses of intacaterol to placebo. Our endpoint is a change from baseline at week 12 in the 24-hour trough FEV1. and we expect the differences from placebo to be between 0.14 and 0.18 liters with a standard deviation of 0.5. We're interested in designing the study for 90% power and an alpha of 0.025. So what I'm going to do is I'm going to initially go now into EAST to see how we can initially try and deal with a design like this. So this is what EAST looks like for those of you who are already familiar with it. The access to the MAMS functionality is through the many samples menu option, either for continuous, for binomial, or for time to event endpoints. So under the continuous, for example, type of endpoints, normal type of endpoints, we have the multiple looks group sequential methodology, which is what we're going to be illustrating today. And that also is the home for the p-value combination approach by Pocharo. This is also the home for the multi-arm module, which I mentioned earlier, which is the fixed sample size, the fixed single stage procedures for comparing multiple arms to control, which I'm also going to be trying to connect with the case study that we're going to be using today. So the first attempt on this is going to be done through trying initially a standard group sequential design, a group sequential design which only has 2 arms and we have 3 looks, right? So by selecting the group sequential design for superiority with 3 looks, we are powering the study for an alpha of 0.025, 90% power, a difference of means of 0.18, which was one of the differences that we're looking at, and the standard deviation of 0.5, which we had mentioned. And for the moment, for each one of the 3 interim looks, we're going to be budgeting our type 1 error using the LAN and the METS version of the O'Brien and Fleming boundaries. And for the moment, we're not going to be using any futility boundary. So if we were to run a design like this, this is a standard group sequential design for 2 arms, this would require a maximum of 329 subjects. So if we were to look at more details about this design, you would see that the maximum sample size is 329, while for a 2-arm trial, we can say 330. So we need 165 subjects per treatment arm. And if the drug works, so under the alternative hypothesis, we would require about 132 subjects per arm. So this is the expected sample size under the alternative hypothesis. And we also have an idea about what our stopping boundaries would look like. And as you can see, at least in the z-scale, when we are visualizing this, since we're using an O'Brien and Fleming type of boundary, it is difficult relatively to stop at the first look. The threshold in the z-scale is about 3.7, and it becomes progressively easier, 2.5 in the second look, and 1.99 in the third look. If we had a single stage design, of course, we know that we would need to compare this to 1.96, right? So this is the group sequential design and how one would deal with that problem if we only had 2 arms in place. In our case, however, we have 3 experimental treatments as well as the control arm. Now, what I'm going to do is I'm going to revisit this problem from a single stage point of view. So if I go into the single look option in EAST, which is a simulation-based approach, this enables us to select different multiple comparison procedures for maintaining the family-wise error rate for a case where, as we mentioned before, we have a common standard deviation of 0.5, and initially we are looking at differences of 0.18 in each one of the 3 arms. So if this is how our responses are going to be generated, then all we have to do is we have to use the Dunnett single-step test as the parametric alternative to the Bonferroni procedure, and then have EAST search through a grid of different sample size values to see what is going to get us close to that 90% power that we are willing to have. So if we were to just have a single stage design, and here I'm just going to try for a sample of size 500, say roughly, I would simulate this design, and then you would see that the design would be simulated 10,000 times, and we would end up rejecting the null hypothesis about 89.6% of the time, which is pretty close to 90% power. So a sample size in the neighborhood of 500 would be sufficient for a single stage design to get us the power that we need for the type of comparisons that we want to do at a single stage. However, where we are now is we want to be using the MAMS methodology, which is not a simulation-based methodology, but as I said, it is an extension of the group sequential methodology. So we have to calculate what the efficacy stopping bounds will look like, and what would be the corresponding procedure if we were to use just one look. So I'm going to attack exactly the same problem, the problem of that I showed before in the single stage approach. I'm just going to have one look here. I'm changing this to one look, and I'm also changing the number of arms to 4, just to have exactly the same setup as the one that I just illustrated. So as you can see now, if I end up putting in the differences of 0.18, 0.18, and 0.18, and I try to use the compute button to see what sample size we would need to get us 90% power, you will see that this would give us 512 subjects. So, and actually, if I went back to the Dunnett test that I showed you before through the simulation approach and entered 512 as a sample size in this simulation, we would actually get at least 90% power. So you can see here, this just illustrates that this method that we're going to be extending now to more than one stages is actually an extension of the Dunnett test. So the natural thing to do is to see what happens if now we have the 3 looks. In the previous case, we have already seen for a group sequential design, if I were to have 2 arms and 3 looks, I would require about 132 subjects on average, right? So now the same thing happens here. I put in 4 looks, 44 arms and 3 looks. I click on my compute button, and then if I look at the detailed output, we will see the following. First of all, the maximum sample size is going to be 520 subjects, which amounts to 130 subjects per treatment arm. If you recall, the maximum sample size per arm in the group sequential methodology for the same targeted treatment effect was 165. Also, if the drug works under the alternative hypothesis, the average sample size is 105. This was 132 in the case that we saw before for the standard group sequential design. Furthermore, we can also visualize the boundaries. I can actually see a graphical approach, which is more informative. If you recall before, we had a boundary of about 3.7 to declare efficacy at the very first look. And at the very last look, we needed a boundary of 1.99. So now instead of 3.7, we have 3.97. It became a little bit more stringent. And that is the same case for the final look, which requires a boundary of 2.4, essentially. So if we were to compare, and this is what I have done here in the slides, you will see, comparing the 2 with the 4-arm boundaries, we have that the higher hurdle to cross with the 4-arm trial compared to the 2-arm trial. You can see that for the different information fractions, the different looks, what the 2-arm boundaries look like, what the 4-arm boundaries look like. These boundaries are higher for the 4-arm trial because this compensates for a greater probability of boundary crossing under the null hypothesis. Okay, you have more chances to cross the boundary when you have 4 treatment arms rather than when you have 2 treatment arms. But the sample size is lower for that 4-arm trial on average because you have more chances to exit under the alternative hypothesis. So the next natural thing to see is what would happen if the values of delta was not the same for each one of the treatments. So if I were to go back to my design here, and instead of looking at a common delta of 0.18 in each one of the 4 treatment arms, I only have 0.18 for treatment 1, while for the other treatments, delta has become smaller. Well, you would expect naturally that with the same sample size as we used before, the power would drop since we have a smaller treatment effect. But if we were to power the study from scratch, now we would require 676 subjects altogether. So now we are at about 169 subjects, which is comparable per arm, which is comparable to what we had before in the 2-arm group sequential design. And correspondingly, under the alternative, the average sample size is about 136. So this tells us that we have the same boundaries as before, but we require a commitment of more subjects per arm, both as a maximum as well as when the treatment works. Now, the 4-arm design requires more subjects per arm than the 2-arm design, but the higher efficacy boundary hurdle is not offset here by extra opportunities to cross the efficacy boundary because only one of the 3 experimental dose levels has a strong effect. Okay, so things go with this design, so like the way that you would think that they would go. So then the one thing that it would be of interest to see now is to explore further these designs and to do that through simulation. The first thing that I did is I added a futility boundary. So I wanted to see what the same sample size that I used before for the efficacy-only approach, which was 676, would buy me in terms of power if I use also a futility boundary with the same Lange-Metz version of O'Brien and Fleming stopping rules. So something that would look like this, where again, in the Z-scale, the efficacy boundary remains the same because we're using a non-binding futility stopping rule, while we have also the opportunity to stop for futility if all the arms cross the futility boundary at the first look. In other words, if the Z statistic is less than 0.09 at the first look, 1.5 at the second look, or of course at the final look we have the same threshold as the efficacy boundary. So with the same sample size, what we have now, if I introduce the futility boundary, we have a drop of about 2% on the power, okay? And this is by introducing the futility boundary. So now we can actually take this design and do a little bit more of it by simulating it. So if you select the design or any design in this, you can click on the simulate button, and that simulate button will bring up a new input screen, which is based on our standard simulation input screen. So here you have the maximum sample size. We have the boundaries that we used for early stopping for efficacy and futility, the way that the responses were generated. Only one of the treatments have a strong treatment effect, and of course, how many simulations you would like to run to see what your achieved power would be if you generate values under this response generation. Well, as we have seen before, the 676 subjects bias roughly 88% power. So if I were to hit on the simulate button, which I can do right now, simulating this design, it doesn't take long, and it would require essentially, it would spit out essentially the same power as the one that we got when we were designing the trial. So what I can do is I can look at the summary of the design as well as the results from the simulation, and I can look at those side by side. And the design itself told me that I would require for the 676 subjects, I would get 88.2 power. While with the simulation-based approach, you can see that we are right on the same area. So what is also reported, so it is the global power, which is the probability that you're demonstrating statistical significance on all of the treatment groups that are truly effective. Sorry, that you're demonstrating statistical significance on one or more groups. But we're also reporting the conjunctive power. So this is if you're interested in demonstrating significance on all of the treatment groups that are truly effective, or the disjunctive power, which is if you're interested in demonstrating significance on at least one treatment that it is truly effective. Along with that, we also have the family-wise error rate, which is the chance of incorrectly demonstrating statistical significance in at least one treatment group, which is truly effective. So this is how simulation is used. Now, if we go back to the simulation screen, however, you will see that we have several other options that will allow us to explore a few more interesting things. And that is something that we can see, first of all, in the boundary tab. When we designed the study, we designed by using what is called the simultaneous stopping rule. We would stop for efficacy if we detect at least one effective treatment. Okay, that was the way that we would stop. However, in the simulation mode, you are also allowed to explore what is called the multiple winner rule. So this is a case where you can identify all of the possible effective treatments within a single MAMS trial. So whenever one or more treatment arms crosses the efficacy boundary, the whole trial does not get stopped, although we stop enrolling patients into the arm that crossed that boundary, but the enrollment and follow-up continues to the rest of the treatment arms and the control arm as before. And then as soon as another one crosses the boundary, then we stop recruiting for that. So we have the opportunity to simulate a multiple winner approach as well. On the same token, you can also simulate different futility options. So the way that we designed the study, we would drop any treatment arm that crosses the futility boundary, and we would stop the study if all of the arms cross a futility boundary. Here, you also have the opportunity to not drop treatment arms unless they all cross a futility boundary at the same look. So there are some modifications of the MAMS design that will allow you to explore different implementations of umbrella studies through the power of simulation. So one of the things that I have illustrated here is I considered a scenario where only one of the treatment arms ended up being effective. The other ones, you can see the difference is practically zero. So what we would expect to see is that the overall power will drop even further. And as you can see here, out of the 10,000 replications that we ended up running, we ended up declaring efficacy 78% of the time. So from 88% power, we have dropped to 78% power if only one of the treatment arms is effective. Furthermore, you have additional details in the simulation sheet. So for example, you can see that treatment 1 would gain you a 77.8% power alone by winning alone, while you would have only 0.47 for treatment 2 and 0.01 for treatment 3. You also have an opportunity to see what the marginal probability of the trial termination by the treatments in each look is. When you're looking only at treatment 1, treatment 2, and treatment 3, as well as what is the chance of stopping early due to one treatment alone at the first look, or to stop at looks 1 or 2 due to one treatment alone and due to any of the other treatments. The simulation-based capabilities of EAST also allow you to validate and check that your type 1 error is being preserved. Because one of the types of simulations you can run is a simulation under the global null. So if you assume that all of the deltas are equal to 0, and then you hit on the simulate button, this will produce again the achieved power for this scenario. But now what we're doing is we want to calculate the probability or the relative frequency of rejecting the null hypothesis when actually the null hypothesis is true. And in this case, what you can see is that when we run this design, we will get, when we run the simulation, we get that out of the 10,000 replications, we end up rejecting 230 times, which gives us a probability or an estimated alpha of 2.3%. We knew ahead of time, of course, that our alpha is being maintained because we are using an alpha spending function, but this also is a way to validate that through the power of simulation. And also now through the updated guidance of the FDA on adaptive designs, the simulation approach is also an acceptable approach of showing that your type 1 error is maintained. Of course, here we also have the opportunity to see it we also have, of course, the property that this is true due to the nature of the stopping function that we're using. Finally, an option that has been added into the software, which allows it also to be comparable to the approach by Posero, the p-value combination approach, is the treatment selection approach. If I go back to this simulation worksheet that I had before, but this time I only stop for efficacy instead of utility, what I can do is I can click on this treatment selection icon, and this will bring up a treatment selection tab on your simulation sheet. So this provides you with 3 different dose treatment selection rules to either select and graduate into the next phase the best number of treatments, R treatments. Here I'm going to try with R equals 2. Select treatments within epsilon of the best treatment, or select treatments greater than a threshold Z, where Z, epsilon, and R are acceptable inputs by the user. So for our example here, what we can do is we can suppose that 2 best treatments are being selected based on the estimated treatment effect at the first interim look. And then if I run the simulation, what I'm going to see is the following. So the first of all is that we can see that our power, the trial has about 89% overall power to detect significance on at least one treatment group, with an average sample size of 466. It also has a chance of about 54% of stopping early by the second look. This is the sum of these 2 probabilities here. You have about 3.5% chance of stopping early on the first look, and about 50.6% chance of stopping early in the second look. So about 54% chance of stopping early by the second look. From the third table, the table of additional details of probability of trial termination at each look, it can be seen that the trial has about 51 or about 52% power to show significance on only one treatment, about 37% to show significance on treatment 2, and less than 1% on treatment 3. And moreover, marginally, treatment 1 has a 65% chance of success, treatment 2 has about 30.5% chance of success, and treatment 3 has about 30.9% chance of success. So through the power of simulation, you always have, of course, the analytical calculations on the design, but through the power of simulation, you can actually perform direct comparisons with other known designs and also explore variations of the original MAMS design by looking at multiple winner rules or looking at rules where you don't drop an arm when one arm crosses the futility boundary, but you only drop if several arms cross the futility boundary at the same time. So all of these have also been shown and have been made available in the different screenshots that I have created in the presentation that you will be presented with. And I hope that all these help you conclude that having a common control arm does save sample size, it reduces the administrative burden, and now we have a common platform instead of trial-specific infrastructures. We continue using the same machinery, even if adding new arms, and it is a much more attractive option for patients, and hence speeds up enrollment because a subject going into the study will have bigger chance getting into an experimental treatment. So overall, it gives you a more efficient use of the resources. These are the references that I mentioned today. So the Posh and Bauer paper of the two-stage combination approach. And then the MAMS methodology is based on work, at least the implementation that you see in EAST, of Pranab Ghosh, Lingyu Liu, Pralay, Gao, and Cyrus Mehta have presented in their 2017 paper. With that, I would like to thank you for attending today's webinar. You can obviously always shoot any questions that you have on the topics that we saw today my way through email. But of course, now we have an opportunity to open up and to submit your questions on the chat window, and I'll be more than happy to entertain them.
Thank you, Pantelis. Before we move into the Q&A, we want to take a moment to remind you that EAST gives you access to a wide selection of trusted, fixed, and adaptive trial designs. EAST is used by nearly every major pharmaceutical company and the FDA. With a broad selection of popular designs in an easy-to-use format, we can help you quickly create and compare trial designs. Our company also offers a range of services from staff that act as an extension of your team and consulting services for more cutting-edge projects to our new real-world analytics capabilities. We are here to help support your objectives. So now we're going to begin answering the questions submitted during today's presentation. As a reminder, you can still submit questions through the questions pane in your attendee control panel. So let's take a look at our first question here. So Pantelis, someone would like to know how this current method compares to the p-value combination approach of Posh et al.
Right. So good question. So I mentioned that first of all you can perform head-to-head comparisons because with the Posh et al. Methodology is entirely simulation based, much like multi-arm is. So what you would have to do is you would have to try out different sample sizes and see what those sample sizes would buy you. You have only one interim look, and then at that interim look, you have the option to advance different treatments into the second stage, a different number of treatments, and you can also stop the study early for efficacy and futility. So in the current implementation that we have in EAST, the Posh method also allows you to perform a sample size re-estimation. So it allows you to increase your resources if your results of the interim are promising. This is something that we're working also for the group sequential extension. Other than that, there is no sort of like a universal winner in terms of of the two approaches. What you would have to do is essentially you would have to try different scenario for the case that you're interested in with one or the other. You can do head-to-head comparisons because the group sequential approach has the ability now through the power of simulation to perform a treatment selection. And then you can constrain it, of course, to just one interim look and see head-to-head for the particular case that you have in mind, who the winner, quote unquote, is going to be. It's not, but there is not sort of like a universal winner or universal solution that provides you best approach.
Thank you, Pantelis. We have one other question here. Someone would like to know with this module if it's possible to recalculate the sample size.
With the module, it is. So the module MAMS, as we mentioned, has two methods. One is the one that we illustrated today, which is the group sequential extension, and then the second one is the p-value combination approach, which the Posh et al. Approach, which has the ability to perform a sample size increase. This is done through the promising zone methodology that was developed by Cyrus Mehta and Stuart Pocock early in the previous decade. And but we are also working on adding this functionality on the group sequential extension. For the moment, MAMS has that functionality, the sample size re-estimation, recalculation, through the p-value combination approach.
Thank you, Pantelis. And it looks like we have one other question here. Well, a couple other questions have just popped up. Can this MAMS module or another module in EAST conduct flat form trials that add a new therapy during during the trial and drop the control due to standard practice and make the super arm the control?
Not currently. So this is not a functionality that we have currently. We are, however, working with a major pharmaceutical company to provide a simulator for such designs. So this is a product that will be you will be hearing about towards the end of the year. But currently, EAST does not have the functionality of adding such arms now.
And we have one last question here. Can you comment on increases in efficiency of the MAMS design in real-life study situations?
I'm trying to see what sort of there are, as I said, in terms of efficiency, there are different ways to quantify it. And I think this is something that I try to show on the very last slide, because efficiency can be sort of like quantified in terms of your chances of and your increased recruitment of subjects, speeding up of enrollment, and therefore minimizing your trial time, increasing the chance of success of your study, which is something that gets achieved by having a common control and comparisons to that. So in many aspects, yes, this is something we have an efficient use of resources. And as I said, in different scenarios, you can also see how well this procedure does compared to some of the other procedures that have been out there longer, like the p-value combination approach, and have at least sort of like a couple of procedures or alternative designs in your kit bag when going in your regulatory talks.
Great, thank you, Pantelis. I think that is it for questions. So before we conclude this presentation, I'd like to show everyone our schedule of upcoming webinars and remind you to register for any of these upcoming webinars that you may be interested in listening to. And if you'd like a certificate of attendance, you can request that via the survey that will come through the thank you email that we will be sending after each webinar. So thank you. Oh, please, Pantelis, if you'd like to comment.
So next week, yeah, we're gonna cover phase 1 dose escalation with, again, a relevant case study, published case study, and hopefully we'll keep your interest in these methods.
Thank you, Pantelis, and thank you to everyone for attending today's webinar on adaptive umbrella trials using the MAMS module. On behalf of Cytel, thank you for joining us today and have a great rest of your day.